Hello students, welcome to Leela's tutorial. So today we'll discuss about protoplast fusion. In last class, we have discussed how we are going to isolate the protoplast from the plant cell and how we are going to culture them in vitro. Right? So first let us see the structure of plant cell where it consists of a rigid cell wall. Then you have the plasma membrane and all the cell organelles and the nucleus. Right? So how you define the protoplast? Protoplast is a plant cell without cell wall. If the cell wall is removed, then you call that plant cell as protoplast, where it consists of all the cell organelles along with nucleus covered with plasma membrane. Is that clear? So let us see what is the use of this protoplast fusion and by what techniques you are going to fuse them. Okay. Before that, what is the use? Why we have to fuse the protoplast? If you are simply isolating the protoplast and producing the plants in plant biotechnology or in plant tissue culture lab, the plantlets will not have any beneficial character, right? They will be same as their parental plant. But here, by protoplast fusion, you are going to fuse two different plant varieties or two different plant species, and then you are going to produce a hybrid plant. Okay, so why we have to go for this protoplast fusion when we have natural pollination process, self-pollination or the cross-pollination? Because here the protoplast fusion we mainly do in between two different plant species, right? Because in two different plant species the natural way of pollination is not occurring, right? It doesn't occur. Right. So here what we do, we isolate the protoplast from two different plant species and allow them to undergo fusion to produce a hybrid cell. Okay. Then that hybrid cell will be cultured in plant tissue culture lab to produce an hybrid plantlet. Okay. Let us see that in detail now. So first it is the fusion of isolated protoplast of two different plant species to produce a somatic hybrid plant. Okay, so it is of two methods. The protoplast fusion is of two methods. First one is spontaneous protoplast fusion and second one is induced protoplast fusion. So the name itself implies the protoplast fusion occurs suddenly or naturally. Okay, and the second one is induced protoplast fusion. That means here we are inducing them to fuse, right? You are inducing them, that means you are either chemically inducing them or with some pressure, external pressure. Let us see that. See, now induced protoplast fusion is again classified into two types. First one is mechanical method and the second one is chemical method. Okay, so in the mechanical method you have only one method, it is by using micropipette. So in micropipette, what you do? Simply you take the protoplast of two different plant species. This may be one and this is the two. Okay, protoplast is isolated from plant species 1 and protoplast from plant species 2. So you isolate the protoplast and what you do? You allow them to add them in a mixture, right? So now you have all the protoplast in that solution which consists of protoplast 1 and also protoplast 2. So with the help of your micropipette, right, with the help of the micropipette, what you do? Simply take the solution so that all these cells, that is cell 1 or the protoplast cell 1, protoplast cell 2, all get entered into the tip of the micropipette. So what you do? You partially close this one, block this one. Okay, partially block the tip and with the pressure release that. So what happens? These, due to the pressure, right, the protoplast 1 and protoplast 2 are forced to fuse. Okay, so that is called as your protoplast fusion by mechanical method. Then you have your chemical method. In case of chemical method, you are going to use the chemical agents, right? The agents which are used in protoplast fusion are referred as fusogens, right? They are referred as fusogens. So these fusogens or chemical fusing or inducing agents. The fusion inducing agents are of 4 types. Okay, mostly in plant biotechnology, we make use of this polyethyl glycol. Okay, so here based on the cell, we select the chemical or the fusogen. Okay, fusogen is the chemical substance which forces or allows the protoplast to get fused. Okay, the first method is polyethyl glycol treatment. So simply by suspending the plant cells or the protoplast 1 and the protoplast 2 in polyethyl glycol, that chemical induces the protoplast fusion. Okay, otherwise simply we go for this calcium cation at high pH. Okay, then other one is aggregation mixture, where we'll be using 10% sucrose and 5.5% sodium nitrate. Okay, in that mixture we'll be allowing your protoplast 1 and protoplast 2 cells to get immersed. So because of that chemicals, here the protoplast come and fuse and they form the hybrids. Okay, and the last one is the electric field. So here very low electric impulse is given in the mixture of cells so that they are forced to get fused. Is that clear? So let us see how the process occurs. So either it is a mechanical or a chemical, the fusion is same and also the formation of somatic hybrid is also same. Okay, so let us see here with the example of polyethyl glycol. So that means here the protoplast fusion is by using fusogens. Okay, so first take the protoplast, isolate the protoplast, select one plant. Okay, first select one plant and select another plant A and another plant B. Okay, from the plant A you are going to isolate the protoplast. So I have named as protoplast A, and from the B plant you are going to again isolate the protoplast, that is protoplast B. Okay, so simply I have differentiated the nucleus. Okay, so here now I want protoplast A cell to fuse with B cell. So what is the use? To form a hybrid. So in the hybrid cell, what I will be getting? I will be getting whatever the beneficial characters are present in the cytoplasm of both the plants, and also the beneficial characters of the nucleus of the both the plants can be obtained in hybrid plant. That means here these plant A may be having certain beneficial characters that may not be present in B, and B may be having certain beneficial characters that may not be present in species A, right? So what I do, I allow this cytoplasm and the nucleus to fuse, so that the hybrid protoplast whatever I get, it consists of beneficial characters of both plant A and also plant B species, right? So here first I am going to isolate the protoplast, then I am going to allow them in a container consisting of PEG, that is polyethyl glycol, right? So after incubation you can see that the protoplast A and the protoplast B cells come closer. Then gradually the plasma membrane present in between both the cells will get dissolved. Okay, as a result there is mixing of cytoplasm, then followed by the fusion of nucleus, right? The cytoplasmic fusion and also the nuclear fusion occurs. As a result you can see now all the cells present in that may not get same proportion of PEG, right? So what may happen, there may be some unfused cells, right, and there may be some fused cells, right? So what you do, you have to allow them to get sedimented. Once they get settled down, you are going to centrifuge them. So based on the density, you are going to separate the somatic hybrid cells and the unfused cells. Okay, so once you go for the centrifugation, the pellet, as the hybrid cells are having more density, they go and settle at the bottom, and in the superficial the unfused cells may be present. So what you do after centrifugation, you are getting the somatic hybrids, right? After fusion you are getting the somatic hybrid cells, and also unfused protoplast may also be present. So simply by washing, the supernatant unfused protoplast cells are washed out and only these somatic hybrid cells are collected. Okay, as a result you can see here the somatic hybrid protoplasts are obtained. Now these somatic hybrid protoplast cells consist of beneficial characters present in the cytoplasm and nucleus of both the plant species, that is plant A and plant B beneficial characters are present in this hybrid protoplast. Now these hybrid protoplasts can be cultured to produce a somatic hybrid plants. Okay, we will discuss how you are going to produce that in next class. For further classes, do subscribe.